Okay, some of you may have started going to prepare for graduation. So we will just proceed, okay? I will record this and later can catch up. All right. So we will look how product concentration can reduce the rate of reaction later, right? So if enzyme have activators, so enzyme also have inhibitors, okay? So inhibitors are molecule that binds to the enzyme and decrease its activity. So inhibitors can be non-specific or specific. So non-specific meaning it can generally cause the denaturation of the enzyme by changing, for example, the pH or the temperature of the surrounding and cause the enzyme to denature. Okay, for example, heavy metal, salts, acids, alkalis, okay, phenols. Okay, some inhibitors are specific, meaning it doesn't change the environment of the enzyme, but it targets specifically to the enzyme. Okay, so usually these are a reversible reaction. They can be the mechanism of specific inhibitors. They can be competitive or non-competitive, alias allostery or feedback. Okay, and they also have some inhibitors are also irreversible. Okay. Okay, so this is just the chart. Okay, so for competitive inhibitor, okay, so under specific we have competitive inhibitor. So competitive means it competes, the inhibitor will compete with something. Alright, so who will this inhibitor compete with? Okay, so this inhibitor will compete with the substrate. So this inhibitor usually resembles the chemical structure and also the molecular geometry of the substrate. Okay, again, this is the structure of enzyme. So we have substrate. So this is the substrate, the blue color one. So what will this inhibitor do? Is usually this inhibitor will have the same shape of the substrate so that it can bind to the active site. Okay. So competes for the same active site as the substrate molecule, and they can interact with the enzyme or no reaction can take place. So sometimes this inhibitor can come and bind and causes some reaction to take place, but sometimes this inhibitor just come and sit at the active site just to prevent the substrate from binding. Okay, so it's either two. And so this reaction has its effect reversed by increasing the substrate concentration. So meaning that, for example, in reversible reaction, whenever the inhibitors sit in the active site, so how do we remove this inhibitor from this enzyme is when we increase the amount of substrate. So it's like creating a pressure to the creating stress to the inhibitors. Okay, so when we increase the amount of substrate, so eventually this substrate can kick off this inhibitor and bind back to the enzyme. Okay, so that is for competitive inhibitors. Okay, something like this. Okay, so we have an enzyme. So this is the substrate. So the competitive inhibitor usually mimics the site where enzyme can, the substrate can bind to the enzyme. So it will fit into the active site. Okay, so how to reverse this reaction of inhibitor? Increase the amount of substrate. Okay, so that is for competitive inhibitor. We also have non-competitive inhibitor. Okay, so if in competitive inhibitors, they will compete with the substrate. How? By binding to the same active site. But for non-competitive inhibitor, they will bind to other side of the enzyme. Okay, okay, let's draw again. This is enzyme. So rather than binding to its active site, maybe the inhibitors can bind to other parts of the enzyme. For example, over here. Okay, and upon binding of this inhibitor to the enzyme, although it doesn't bind to the active site, it can still change the shape of the enzyme, causing the conformational change to the enzyme. So meaning that when this inhibitor bind to this enzyme, suddenly now the active site is changed. Okay, Because remember, enzymes are proteins, okay? Okay. So if the inhibition is at place remote from the active site, it is called allosteric inhibition. Okay, so non-competitive inhibitors, one of it is the allosteric inhibition, other parts than the active site. Okay, so the interaction of an inhibitor at an allosteric site changes the structure of enzyme. So the active site is also changed, cannot have its effect reversed by adding more substrate. Alright, because now the active site is already changed, so no point in adding more substrate. Okay, this is the example. Okay, so in normal reaction, okay, substrate will bind to the active site and then turn into product. Okay, but in the non-competitive inhibitor, so enzyme has it inhibitor site, or what we call as the allosteric site just now. So this inhibitor will bind to the allosteric site. So observe now, the active site is changed. So now substrate cannot bind. Okay, in some cases, substrate can still bind, but there's no reaction that takes place. Okay, because the structure of the enzyme is changed as a whole. All right. So in this condition, even though we increase the amount of substrate, nothing will happen because the enzyme is already changed. Okay, so this is another representation for it. So this is competitive inhibitor binding to the same active site as the substrate, but for non-competitive, it will bind to the allosteric site of the enzyme, but still they are able to change the active site of the enzyme, preventing the substrate from binding to them. Okay, in some cases, the inhibitors are irreversible. Okay, meaning that they can covalently bind to the side chain of the active site. Okay, we don't have the diagram over here. Okay, so in non-competitive inhibitors, sometimes it can be reversed, but for irreversible inhibitors, they usually will change the side chains within the active site, meaning it cannot be turned back into the normal or physiological structure of the active site. Okay, for example, aspirin. So aspirin will covalently modify the key enzyme involved in inflammation. Okay, when someone is having fever, for example, doctor will prescribe, for example, aspirin. Or someone is having allergic, so doctor will prescribe aspirin. So this aspirin will covalently bind. So if you remember, covalent bonds are one of the strongest bonds. So when this aspirin binds to the enzyme involved in inflammation, the enzyme can no longer facilitate inflammation process. Okay, because aspirin has been irreversibly changed the structure of the enzyme. Okay, we also have feedback inhibitors. Okay, this is when the amount of product will affect the rate of reaction. Okay, this is quite common in our body. So in the human physiology, we always have this what we call as feedback loop. Okay, meaning that the end product of something will stop the initial reaction. Okay, for example, if we have a sequence of reaction from initial product A to B and then finally G. Okay, and each reaction is facilitated by a type of enzyme. Enzyme 1, enzyme 2, enzyme 3, and so on. Okay, so what will stop A from keep on turning into B and C and D is the amount of G. Okay, so this is what we call as the feedback mechanism. When the amount of G increase, it will send signal to A, or sometimes in certain reactions, G will directly bind to enzyme 1 so that A cannot bind to enzyme 1. So this will stop the reaction so that we can control reactions in our body. Okay, we won't be having this like infinity reaction in our body. At some point, okay, the product itself will cause the reaction to become plateau to control the amount of product that is formed. Okay. Okay, so product is used in the next step in the pathway. So the final product will become the inhibitor of the earlier step. So that is feedback inhibitor. Okay. All right. So that is all about enzymes for today. Do you have any question? No. All right. All good. Okay. So this is more like a recap. Okay. We will find a lot of enzyme reactions or biochemical reaction later on. So if you are confused or, or whatever, you can come back to this chapter later. Okay.
Doctor, can I ask you a question?
Yes, Miss Lim.
How can we know whether we have sufficient enzyme in our body or not?
Okay. So that is something that for us not to worry about. So usually the amount of enzyme would be sufficient. As long as you are healthy, you are feeling good, then meaning all the reactions in our body is taking is at its optimum rate of reaction. Okay. Uh, so the indicators showing that there might be some disruption in terms of our enzymes. Okay, there's damage to our enzymes. So there are certain diseases that is related to enzymes. Okay. For example, the common symptoms would be when we had fever or we have trouble in digesting certain things. So certain people, they are lactose intolerant. Okay. So whenever they drink milk, they keep on vomiting, for example, or they are experiencing diarrhea. So that's one of the indicator. Okay, there might be something wrong inside that person's body that's making them unable to digest, for example, things inside the milk. So in lactose intolerant people, they don't have the enzyme lactase. Either they don't have or they don't have sufficient or the enzyme is alterated, for example. Okay, that's making them unable to digest lactose inside the milk. That's why whenever they keep on consuming milk, they will vomit, for example, or having diarrhea or some certain allergic reaction. Okay, so that's an indicator they don't have lactase or they may have insufficient lactase, the enzyme lactase. So that's how we know. So generally, if you are healthy, we can eat almost anything. That's usually fine lah, the amount or the enzyme condition in our body.
Oh, okay. Thank you so much.
Okay. So any other question? Okay, so that's why it's always, so remember when we talk about factors affecting enzyme activity, that includes temperature and also pH. So if you have fever, especially high fever, go check, don't just like let it be, because high temperature inside our body can disrupt the enzymes reaction, may denature our enzyme. And another second thing that I would like to highlight is when we are taking something, for example, supplements that we are taking or any products that we are consuming, please be very detailed in checking the ingredients, whether or not those ingredients can change or alter the pH inside our body. Is the things that we are consuming making our body more acidic or more alkaline? Okay, because that would also alter the enzymes inside our body, which can cause to a greater extent failure in organs, for example. Okay. Yes, can PM if you have question. Okay, I will share again the attendance for those who have not signed in. So if you have no more question, then we will just end the class for today. Okay, we will discuss the tutorial questions in the next session. I will let you know when.
Okay, madam.
All right.
Madam, thank you.
Thank you, everyone.
Thank you, madam.
Okay, thank you, Doctor.